Everybody, I think it's noon. I think it's time we need to get started. My name is Jim Lamandia. I'm the chief scientist at the Valley Laboratory, and I'd like to introduce today's seminar. It's going to be presented by Dr. Srikanth Kodati. He's been working with us for 2 years now. Came to us from, he got his PhD at the University of Nebraska-Lincoln, working on the diversity of Rhizotonia species in both crop and native plants in the Sand Hills region. He's been working on 2 projects at the experiment station, Boxwood Blight, and also our hops project. And he's going to present part of his Boxwood Blight data today, talking about some of the practical impacts that we found on epidemiology and how it may impact disease and hopefully eventually management. So let me introduce to you Dr. Srikanth Kodati. I also might mention that he, from us, will be going on to work as a combination of time with the University of Connecticut and continuing his project with Boxwood Blight here at the experiment station. So an example of collaboration, I guess, too. Begin.
Thank you, Dr. Lemondia. I hope I'm audible. Thank you for the introduction. Myself, Srikanth Kodati from Valley Laboratory. I joined in Valley Laboratory in 2020, February 28th, just before COVID kicks in. So today we'll be talking on Boxwood Blight disease and its epidemiology, what we are doing in Valley Laboratory for the management practices. So a little bit about myself. I did my bachelor's in agriculture in Hyderabad. You can see I'm from India. For master's, I came to Tennessee State University. I did my PhD from University of Nebraska-Lincoln. My project was on diversity and pathogenicity of Rhizotonia species that was collected from different regions of Nebraska. So I conducted statewide survey, collected soil samples and plant samples, and from natural plant in Sand Hills also. So I also conducted the pathogenicity tests in grow chamber and greenhouse conditions. It comes to the boxwood. So the boxwood belongs to the genus Buxus. The Buxus means box in Latin. So probably the name was given to this plant because the wood from this plant was used to prepare ornament boxes. So it has been used as religious purposes and preparing the instruments. This hardwood was used in other purposes like printing blocks. Roughly there are 100 species. They grow everywhere except in continents Australia and Antarctica. The most familiar boxwood species is called Buxus sempervirens, which is native to Europe, and the dwarf cultivar from the Buxus sempervirens is called English boxwood in Europe and in North America. So this is most familiar boxwood cultivar among the cultivars available in boxwood. There are almost 400 cultivars made from the species Buxus sempervirens. This plant is used for the hedges, peteris, and specimen planting, and topiaries. So when it comes to the disease, the Boxwood Blight on boxwood, this plant also called box in Europe. We call it boxwood here, boxwood plant, but in Europe, most of the people, they know it as box plant. The first this disease was recorded in UK in 1994. This disease was described and the name was given to this pathogen and the disease in 2002 in New Zealand. So when they conducted the population studies on the blight pathogen, they found that it is not a single pathogen. There were 2 lineages. 12% of the population belongs to the G2. The name was given initially G2, the genetic lineage 2, and the G1 is the majority of the population belongs to the G1. It was named as Cylindrocladium pseudonaviculum initially, and the name was changed to Colnectria pseudonaviculata, and the genetic lineage 2 was called Colnectria henricotiae. Why this disease is important for us? So when we look at the dollar value of this boxwood plant and the economic impact of this pathogen on the boxwood production, boxwood is the #1 woody ornamental plant with the value of more than $141 million in US. And this disease was first found in Connecticut and North Carolina in 2011. Currently, within 10 years, it spread to more than half of the states, 30 states and 3 provinces in Canada. In Connecticut, it caused almost $5.5 million of loss in first 3 years of identification of the disease. So when we look at the specific timing in Connecticut, so in 2011, there are 8 landscapes that were tested positive for the Boxwood Blight. Within 7 years, it rose to 400 landscapes. When we look into a specific month, October 2018, there are 192 landscapes that were tested positive for the Boxwood Blight. What causing the huge infections in specific October? When we look at the conditions in October of 2018, between August to October, we received almost 19 inches of the rain over 37 days. There was the relative humidity more than 90% for 77 days in that period. So maybe the precipitation, the rainfall, and the relative humidity might be causing the highest, the increase in the infections on boxwood. Look at the host range of this pathogen. This pathogen was initially found on boxwood. So the English boxwood, which is the dwarf variety Buxus sempervirens safruticosa, is a highly susceptible one. And host pathogen split tests were also conducted on other hosts like Sarcococca, which is called sweet box, and Pachysantha species. So these 2 species are grown in the same nurseries where the boxwood is grown. So these 2 species also tested positive, tested as the hosts for the Boxwood Blight pathogen. So these plants can be act as alternate hosts to spread the disease onto the boxwood plants when they come close to them. As we know, there are 2 lineage groups. So there is no difference for these 2 lineage groups in terms of host range. So these 2 pathogens can cause almost similar plants. When we look at the infection cycle, when the spore fallen on the leaf, it takes like 3 hours for the germination when the conditions are conducive. After 5 hours, it penetrates through the stomata. Penetration through cuticle is very rare. It will not produce any hastoria or aprosorium to penetrate through the cuticle. It penetrates through the stomata and grows between intercellularly. And after 6 days growing in intercellularly, they would come out from the stomata back and produce the conidia, blooms of conidia. And this life cycle is very short. It will be completed in 7 days. It is ready for the spreading secondary infection. When we look at the symptoms, so the Boxwood Blight symptoms are very distinctive. They can be clearly separated from the other diseases on boxwood. Sometimes it may we may confuse that it may be from the Volutella. So there is a little bit difference between these 2. Initially, they start as a small dark lesion. Most of the times, the symptom starts from the edge of the leaf. Eventually, they grow bigger or coalesce with other symptoms nearby, and they form the bigger lesions. We can see the yellow halo around the symptom, and the border of the symptom is in dark brown. The center of the symptom, the center of the lesion is the necrotic lesions. So these are the specific things we can separate this Boxwood Blight from the other diseases. When the conditions are very conducive, the whole plant can be infected. All the leaves can show the symptom and they can dry and they defoliate. It also infects the stem part of the plant. The stem part shows the dark stripes initially. They grow bigger and the whole stem becomes the dark in color and they produce the conidia also. This is the plant situation when the situations are conducive and the high amount of inoculum present. They can defoliate all the leaves and the stem, it will become like the dried plant. We can see the defoliated leaves on beneath the plant. Here is one of the leaves, the dried leaves after the infection. We can see the bloom of conidia coming out from the stomata. When we observe the conidia closely, it produces in clusters, a white color cylindrical 2-celled conidia, and it possesses the white slime on the surfaces of the conidia. And beneath, we see some stellar structures. They grow bigger than the conidia. It gives the whitish growth to observe on the backside of the leaf. When the conditions are not conducive, hard conditions, so the mycelium inside the leaf would produce the chlamydospores, the resting bodies, which has they are melanized. They aggregate and form the microsclerotia. So microsclerotia. In general, there would be 3,600 of microsclerotia would present in one single leaf. They can survive for 30 weeks in the soil, and they can survive for 30 months in the buried stems and the buried leaves. So they can easily overcome adverse conditions, and they can be source for the infection in the next season when the situation is conducive. So how this is spreads? We saw the conidia, and this is the size of the conidia is larger in size. So the wind dispersal is mostly unlikely. It spreads through the water splashes and rain associated with the heavy winds. And as I just mentioned, when the plant is defoliated, all the leaves will be found on the beneath of the plant. Those leaf debris will be the source of the second inoculation in the next season. So the chlamydospores are the conidia. Conidia can directly infect the plant, but when there is chlamydospores present or microsclerotia present, they would produce the conidia first. Then the conidia would germinate and infects the leaf tissue again. These spores, as I mentioned, it has some slime layer. Because of that, it can stick to the animals or tools or transport tools or animals and the human beings. Like when we are working in the landscape, it can stick to our shoes or pant, and it can spread to the healthy plants. The transport of the nursery stock, asymptomatic nursery stock. So it would cause the spread of the disease from one continent to the distant transport of the infection. What are the conditions that are very conducive for the infection? So when we look at the conditions that are favorable, obviously the planting of high susceptible cultivars, and the temperature is reported to have, like, the optimum temperature is 23.7 degrees centigrade, and no infection is found at above 30 degrees centigrade. High humidity and high precipitation causes the severe infection. As we know that rainfall with the high speed winds could take the conidia to the longer distances and spread the disease to the longer distances. Inoculum concentration also play an important role. When higher level of the inoculum, the disease can caused in very soon, and it can spread very fast. So here is one of the reports that is conducted on impact of interrupted leaf wetness on the infection. So there are two different initial wetness periods, and there was a middle interrupted dry period between the wetness periods. So when we see there are 2 5 hours and 8 hours of initial wetness periods, and half an hour, 3 hours, 8 hours of dry periods, and 12 hours of final wetness periods. Here I'm gonna show few details here. When there is initial wetness period 5 hours and there is no dry period, and 12 hours of final wetness period. So 17 hours straight wetness period caused the high level of disease. When there is half an hour of middle wetness period for the same 17 hours, the disease infection has been reduced drastically. Likewise, here 8 hours and 12 hours of wetness period with no interrupted dry period, the total straight 20 hours of leaf wetness period caused the disease, and with the half an hour of the dry period between 8 hours and 12 hours, it reduced the disease. So a small amount of wetness period also reducing the disease significantly. What is the impact of this blight disease? Here is one of the locations where we received the picture and we received the samples. The total area is infected with the Boxwood blight disease. So there is no plant survived. So as I have mentioned, this boxwood has an historical importance. Most of the historical gardens are planted with the highly susceptible English boxwood. So now it is becoming a problem because of this disease. There is a need to replace the whole boxwood plants in the gardens, or we need to find a better management practices. So this situation is making the landscape owners or nursery owners to move out of the business from the boxwood. So they are going for the other ornamental plants. There are so many we have so many examples of the nurseries who burned all the boxwood plants in their nurseries. So having that information on back in hand, and we developed studies to evaluate the effect of leaf wetness period and temperature on infection and sporulation. And we have developed a model to predict the disease progression. This study initially was conducted by Dr. Elisha Perkins, who was the postdoc who worked in the same lab before I joined. She conducted the preliminary trials using green velvet cultivar, which is known as a moderately susceptible cultivar for Boxwood Blight. And 14 leaf wetness periods were taken for the study between 4 hours to 24 hours. And it was conducted at 18, 21, and 27 degrees centigrade. The number of infected leaves were collected. The data of the number of the leaves collected, infected was collected in at 3 days, 6 days, 9 days, and 13 days post inoculation. Based on that preliminary data, we expanded the experiments to conduct on different cultivars and different temperature points. In the second level, we conducted this experiment at 18 degrees centigrade and 25 degrees centigrade on 3 cultivars. One of Safriticosa, cultivar green velvet, cultivar winter gym. Safriticosa is a highly susceptible cultivar. Green velvet is moderately susceptible one. Winter gym is less susceptible one. For the inoculum preparation, the pathogen was grown on PDA for 7 days at laboratory conditions, and the plugs with the conidium were taken into the distilled water with the 20, and we stirred them, dislodged the conidia, and prepared the spore suspension. So we made sure that each 50 microliters of spore suspension has 200 conidia. So for the process of this experiment, as you can see, each plate has 2 glass slides, and each glass slides contain 5 leaves, detached leaves, and each leaf was, all these leaves were prior disinfected, sterilized, surface sterilized with the 10% bleach, and they were washed with the running tap water, and they were allowed to dry. After drying them, they were placed on the glass slides, and each leaf was inoculated with a drop of spore suspension, which contained 200 conidia. And these leaves were incubated for 9 days at respective temperatures, and at their leaf wetness periods. Per se, if we have 4 hours of leaf wetness period treatment, so we came back to the lab and we let the drop on the leaf run, and we let the leaves dry. We made sure that there is no wetness after 4 hours on the leaf for the specific treatment. And that data was collected once in every 3 days. So here are the findings from the experiment. On the right side, you can see the infection percentage on y-axis. The infection percentage is the number of the leaves infected. On y-axis, we have cultivars: Buxus, Safriticosa, green velvet, and winter gym. And this data is showing from the 18 degrees centigrade and 25 degrees centigrade. And this sporulation data, the number of leaves that show sporulation, the cultivars on x-axis. When we closely look at the data of the infection, the Buxus Safriticosa, as we know, it is highly susceptible, showed maximum amount of the disease at 2 temperature points, 18 degrees centigrade and 25 degrees centigrade. When it comes to the green velvet and winter gym, interestingly, it is moderately susceptible one, it is less susceptible one. But there is no significant difference between these 2 cultivars in terms of the infection at 25 degrees centigrade and 18 degrees centigrade too. And come to the sporulation. The sporulation on Buxus Safriticosa and sporulation on green velvet leaves there is no significant difference. The highly susceptible cultivar and moderately susceptible cultivar, they showed similar sporulation at 18 degrees centigrade. But at 25 degrees centigrade, all these 3 cultivars are significantly different. So we can see here another interesting point that winter gym, it showed high level of infection at both the temperatures. But when it comes to the sporulation has been reduced. It is showing that the least susceptible cultivars, though they are having the infection, they are showing this disease symptoms, but they are stopping the sporulation of the pathogen. Indirectly, they can stop the spread of the disease within the landscape or from one area to another area. Here, I'm showing the proportion of the infected leaves that are showing the sporulation. So on x-axis, we have the on y-axis we have the number of the leaves, on x-axis days post inoculation. So on left, on your right column, we have 18 degrees centigrade one and 25 degrees centigrade on left. So we can compare the Sapruticosa leaves sporulation on at 18 degrees centigrade and 25 degrees centigrade. At 3 days of post inoculation, we don't see any kind of sporulation on 3 cultivars at 18 degrees centigrade, but the darker region shows the sporulated proportion of the leaves in total infected leaves. So each bar indicates the infected leaves, the total number of the infected leaves. The darker region indicates the sporulated leaves. So we can see the sporulation started in 6 days post inoculation and 9 days for both Sapruticosa and green velvet. The sporulation amount in terms of with respect to the infected leaves is reduced for the winter gym. We can see high maximum number of leaves showed the sporulation at 25 degrees centigrade for Sapruticosa at 6 days and 9 days. So at 25 degrees centigrade, even the Sapruticosa leaves showed the sporulation where they did not show at 18 degrees centigrade. And the moderately susceptible cultivar also showed sporulation from 3 days at conducive temperatures. Here we analyzed this data using nonlinear regression, 3-parameter nonlinear regression curve. So this data fit to this formula. Here we have 3 parameters, A, B, and X0. A indicates the maximum expected amount of the disease through this curve. B is the inverse of the slope of the curve. X0 is the inflection point. Inflection point is the point where the slope is maximum. So when we look into the Buxus Sapruticosa data, the A value for both at 18 degrees centigrade and 25 degrees centigrade, there is not much difference. So the maximum amount of difference on highly susceptible cultivars at low temperature and high temperature is same almost. But the X0 values are different. So X0 is it is 7.94, means it needs at least 8 hours of leaf wetness period for taking the high level of infection. At 25 degrees centigrade, it is 6. So at lower temperature, they need high duration of leaf wetness period with compared to the infection at high temperatures at the same cultivar. So this change for the green velvet at 18 degrees centigrade and 25 degrees centigrade is different. So it is causing less amount of disease at 18 and higher at 25 degrees centigrade. This is consistent for the another cultivar too, winter gym. It is lower at 18 degrees centigrade and higher at 25 degrees centigrade. So the X0 values, the reduction in the X0 values from 18 to 25 degrees centigrade is consistent for the other 2 cultivars also. So they are indicating that at lower temperatures, they need a higher duration of the leaf wetness periods for all the cultivars. But the amount of disease developed on cultivars are different. So we conducted a multilinear linear regression analysis to develop a model where we can predict disease progression based on the environmental factors. So on x-axis we have leaf wetness periods. On y-axis we have the days post inoculation. So we have developed six graphs for 18 degree, 3 graphs for 18 degrees centigrade for 3 cultivars and 3 for 25 degrees centigrade. So how we can use this graph? So per se, we are taking a line from 8 degrees post inoculation and 18 hours of leaf wetness period. So this is the point. We can see the estimated disease infection would be 39% on winter gym. At the same conditions on green velvet, the disease infection percentage would be 57. And the same conditions on Sapruticosa, at 5 days post inoculation at 18 hours of leaf wetness period, it causes the 65% of the disease infection. So these graphs would be helpful, and this model is very helpful to predict what would be the disease infection, what disease we can expect in 5 days, and we can take the management practices we can adapt. We can see there is a clear difference between 18 degrees column and 25 degrees column. So there is the curve of the disease infection from 16 to 22 degrees centigrade, there is something going on. There is a reduction of the disease on the leaves. So this temperature and 25 degrees centigrade is very conducive for another biological activity. So that is interfering with the infection of the conidia. So that made these curves look differently. So what are the outcomes from my this experiment, the first experiment? So we found that there is a need of at least 5 hours of leaf wetness period, and the infection has been is increased between 12 hours to 15 hours, and it continued until 21 hours of leaf wetness period. After 21 hours, there is no difference. Though there is wetness, the leaf is wet for more than 21 hours, but there is no difference of leaf wetness period. We found the maximum infection and sporulation occurred at 25 degrees centigrade. It needs 7 days for the, there is a lag of gap of 7 days between the infection to the sporulation. And as I show, winter gem is showing the infection symptoms, but not this sporulation. That is saying that less susceptible cultivars would stop the spread or reduce the spread of the disease. So what are the management practices we need to adapt? So we need to remove the diseased plants, remove all the debris, not by sweeping, because it can infect the other healthy plants. So it is better to do it by vacuuming or just sweep each plant individually so that it cannot, the spores cannot travel. Sanitizing the tools frequently is required, and pruning should be done at a dry or low humidity conditions. This is important. So this is one of the examples we got from the people. We got the samples, and we see that this is infection on the plants. Though they practice all the best management practices reported or explained, what causing it. So we conducted a small experiment by scraping the leaves onto a cover slip, and we incubated for 3 days, 6 days, 9 days, and 12 days, and we transferred them onto the PDA to see if the conidia is survived for 9 days or 12 days. Interestingly, we found that they can survive more than 12 days when they are clumps. So we wanted to see the conidia, how many days they can survive at different levels of humidity conditions. For this experiment, we conducted initially at two humidity points, 2 humidity conditions. One is dry and the wet conditions. So I hope you can see there is a glass rod, and we have 2 microscopic slides, and I have 4 cover slips on each slide. So there are 8 cover slips. So each cover slip receives the treatment of conidia in 2 different ways. So initially this experiment was conducted in 2 conditions, wet condition and dry condition. For wet conditions, we added warm water to the humidity chamber, and for the dry conditions, we added silica descent. After experiment 1, we found significant findings. Then we wanted to expand with the different humidity factors included. So we included 80% of humidity by adding the saturated potassium nitrate solution in the bottom, and we achieved 40% humidity by adding saturated magnesium chloride solution. So these are the findings from the individual conidia and the clumps. The relative humidity at 100 degrees, 100%, we calculated the percent of individual conidia germinated and clumps of conidia germinated. The individual conidia, the percentage we calculated each cover slip under the microscope at 110 magnification, 110 times magnification, and we counted 100 spores, at least 100 spores from the each microscopic slides. So these are the 14.1 is the average of the microscopic slides which showed the conidia germination. So at 3 days post inoculation, individual conidia showed 14.1%, and the clumps 37.7%. At 6 days, 9.3% and 27.2. So when we look at the individual conidia, they all germinated until 9 days of post inoculation at 40% of relative humidity also. But at 15% relative humidity, the germination was observed in individual conidia at 3 days post inoculation and 6 days post inoculation. But there is no germination observed at 9 days post inoculation. When it comes to the clumps, the germination was observed at all relative humidity points and all days post inoculation. So here the I saw, so each cover slip was considered as a replicate. So there are a total of 16 replicates for each treatment. So here we see this data is showing the positive and negative. So if there is some germination in one cover slip, it is considered as the positive. So if there is no germination on the cover slip, it is considered as the negative. So when we look at the germination on the cover slips at 100 relative humidity points, there are 13 out of 16 replicates showed the germination. Some germination, at least one conidia germinated on cover slip out of 100. When it comes to the clump, there are 100% of the cover slips. The 16 cover slips showed the germination. At 18, the germination has been reduced to 12, and 40, the individual conidia germinated in only 7 cover slips out of 60. So 80%, more than 80% germination was observed at 100 relative humidity. At 40, it became to half. At 15% relative humidity, the germination percentage is 0. But in clumps, 100% in all conditions. So there is no effect on clumps with the relative humidity at days post inoculation. So what we found from this experiment? So the germination was observed at 3 days and 6 days post inoculation in dry conditions for the individual spores. For the clumps, there is no difference. They can germinate until 9. If we conduct it for the 15 days, also, if we expand that, I think we could find the germinated spores after 15 days too. The significant difference between individual and clumps germination in dry conditions. So when we are doing the pruning, most of the times the conidia can attach to the pruning tools and other things in clumps only, because they produce in clusters. So the slang could help them to stick to the tools, and there is no significant difference for the clumps germination at any temperature point and at any days post inoculation. So these tools can be the source of the infection for the healthy plants. And there is no difference humidity conditions on the germination. So even it doesn't matter if they are dry conditions or the wet conditions, these spores can still survive for 9 days and they can infect after. The number of days exposure has no effect. Humidity has the significant effect on the individual spores. So the practical implications, the infection is higher at conducive at 21 to 25 degrees centigrade. We need to regularly scout the plant to identify the diseased plant so that we can remove them soon so that it cannot infect the other healthy plants. Buying the healthy plants from nurseries is recommended, and sanitize the tools frequently from one landscape to another landscape, or from pruning the diseased plants to before moving to the healthy plants. Planting the less susceptible cultivars is better so that it can stop the spread of the disease. And we need to modify the best management practices. As I showed in other best management practices, they recommend us to do the pruning in dry conditions, but this experiment, the second experiment, is showing that the dry conditions may not help to stop the disease. So we need to remodify the best management practices and change the conditions. I'd like to thank my advisor, Dr. Lamonde, and other funding agencies, USDA, State of Connecticut State's Department of Agriculture, and HRA for supporting this project, and the technicians from Dr. Lamonde's group for helping me to conducting these experiments. And Elisha Perkins, so she initiated the project first, and that preliminary data really helped me to form the other objectives and conduct the complete experiments. These two experiments, I prepared 2 papers from these 2 experiments, and one of them is already accepted for the publication. Another one is under the review for the APS journals. I would like to take any questions you may have. Thank you. Sure. Yes.
Could you talk a little bit more about the asymptomatic nature of the disease? I mean, is that you see a significant amount of that? How transmissible is it?
Okay. So the impact of using transferring or buying the asymptomatic plants from the nursery, how it could impact the disease spread from one location to another location. Right? So this disease takes like 3 days to show the infection symptoms on the leaf. So though there is infection on the leaf, we may not see the symptoms for 3 days. If you buy any infected asymptomatic plant, it already has the source of the pathogen in it. So once you move the plant to your locations, or when you buy asymptomatic plant from nursery, you take them to your location and you plant it. Within 3 to 4 days or 5 days, it starts showing the symptom and it produces the spores. So you are bringing the inoculum, you are bringing the pathogen to your location when you are buying the asymptomatic plant. So it is better to be careful. And we need to observe carefully, because this disease initially starts from the leaves inside the plant, inside the canopy, not from the outside leaves. So it is we can easily deceive that this plant is very healthy. So we need to carefully observe the leaves inside too, so that we can avoid buying asymptomatic infected plants. Sure.
So you showed that the sporulation is affected by the relative humidity with 180 and 60 and 40. So considering in the reality conditions, we constantly observe there's a fluctuation in the day and in the night. During the night, they're usually like about 90% in the summer, and in the days it could be like 40 or 50 depending on the weather. So during these fluctuations, do you predict some of those events are happening more in the evening versus in the day, or?
So the question is, the relative humidity is influencing the development of the amount of spores germinated in the infected plant. So there will be a fluctuation of relative humidity in the daytime and nighttime and of season to season. So so the question is if we can predict based on that, right?
Like you expect in the evening there will be more sporulation than the day.
Yes. So this sporulation is highly impacted with the microclimate nearby the plant. So when the plant is irrigated, so most of the times the change is smaller with compared to the other environmental factors. So it would be, we can expect that there would be a high amount of sporulation when the relative humidity is higher in the night times. So that the longer time we have the high humidity, when we can expect the more sporulation. I hope makes sense.
Giant.
Sure.
So I think what you're asking is about survival, right? So the spores germinate when they have free moisture, when there's and the experiment that the second experiment he was describing is what is the survival when it's dry.
Okay.
Right. So what he demonstrated was that whether it's 15% moisture or 95 or more, I mean humidity without free moisture, so you don't get germination, can the spores survive dry periods, and then when they get conditions suitable for infection, germinate and infect plants. And he demonstrated that is indeed happening. And I think that's to tie this into what the previous question. Sometimes plants will come from areas where they're produced under dry conditions and have very little or low levels of disease on the interior of the plant, but maybe have these sticky spores on the leaves. They'll survive days or maybe weeks until they get to a location where they get wet conditions and 8 hours of leaf wetness, and then you can get infection. So this actually totally upends those best management practices, assuming that you can just prune when it's dry and you're not going to get infection. You can prune, spread it, and a week later, when it gets wet, you can get infection.
Sure.
You have already explained this in my use. So does the pathogen structure require plant? Because we have to be aware. Maybe because
So the question is if we have any survival structures or survival spores in the adverse conditions in the winter for one season to another season. Yes, so this pathogen can survive as microsclerotia in the infected leaves and infected twigs, and they can survive in leaf debris for 30 weeks in the soil. And if they are within the leaf or within the stem parts, they can survive for 30 months in the adverse conditions. So they can form conidia, and then that conidia can infect the new generation plants. Right. Yeah. Sure. If it is not already, the question is if there is only one boxwood plant in our backyard, so how we can save that plant? If it is not infected, we can regularly check if there is any infection coming. If you find any infection before it starts sporulating, better to remove that twig or plant part and discard it. And once the infection is when the plant is infected and it's sporulated, we need to clearly monitor or take the measurement practices for few seasons to remove the inoculum dose from your location. Fungicides will help, but they can only protect you from protect the plant for some time, not the complete protection. They cannot give you the complete protection. And we are also working on different management practices, looking at the different chemicals and the biologicals that can impact the disease management in future.
I had one question. When the outbreak first happened, it caused a lot of damage, and then we had a few years there where we didn't see it. What's the current strategy as to where was it surviving in a microsporulation form during that period, or was it just a low smoldering infection in the interiors of these canopies?
So for the survival of the question is if there is a break for the infection some duration of the time. So they can survive as we saw that there are some alternate hosts, and this plant can infect the lower leaves also if there is the conditions are conducive. But for the survival for the longer duration, the chlamydospores are the microsclerotium is the main source. They can survive for longer duration when the conditions are not good. Like if we see continuous dry conditions and non-rainy season for a significant amount of time, it will not show any significant disease, or they may not produce spores for the secondary infection.
So wait, one of the issues that we can have with this is that if you've got a boxwood plant and if you have boxwood in your yard, and when we evaluate disease, if you try to look at all the leaves on a boxwood plant, I mean, there are thousands of leaves, and the reason they like boxwood for topiary is because the leaves are retained right into the center of the plants. So if you've got a plant that's got 1 or 2 lesions on either a stem or a leaf on the interior of that plant, good luck trying to find it. And under the first year that this showed up was our wettest year in history. 2018 was the second wettest year in history, and there's no surprise that those 2 years were the ones where the disease really flared up. So once you give it the right conditions, it goes from having this that's a great example of a smoldering fire to this conflagration. It just takes off and goes through the plant. So whether it's in the nursery industry, where it's being shipped from somewhere with 1 or 2 lesions, or whether it's in a landscape with 1 or 2 lesions, that's in most diseases that's inconsequential. But with this one, when you give it good conditions, it really goes crazy.
Jason.
So I'm not a plant person, but you mentioned the range of tolerance of the different cultivar varieties. Is there any understanding of the mechanism of that tolerance? Is it action or better able to deal with it?
There might be, so what would be the source of resistance or the susceptibility from the different cultivars? That is the question, right? So we have not looked deeply into it, but there will be, there is a difference. So the mainly this conidia would penetrates into the tissue or into the leaf through stomata. So there will be a difference in number of stomata for each leaf, that thickness that can penetrate into. or there are some other factors too, but we have not looked into it yet. There is another interesting project we can look into to see what are the leaf features that is helping to stop the reduction in the infection. Sure.
It's a little off topic, but do you know anything like a dry eye strategy?
Control, sorry, control what? The question is if I know anyone using the RNAi technology to reduce or to stop the infection, to stop the pathogen growing inside the plant. So as that is interesting area to work with. Actually, we wanted to work in some part of the time. I and Dr. Lemondi and Dr. Coles from a Valley Laboratory, we brainstormed something to do with this, but we did not get the chance to go further with that. I don't know anyone who is working on that topic yet. So that is an interesting area to generate the resistant cultivars.
I think that'll be a nice candidate.
Sure. Yeah, sure. Yeah.
Any other questions? Thank you.
Thank you so much for your time for listening to me.